Yeah.
So I have one question and no, not one, but three questions
Mm-hmm.
in the chat box. The first one, how do Sophia solution compare in noise reduction efficiency versus the other vendor like Illumina Dragon, Sentient, or GetK best practice? So can you give more detail to
Okay.
elaborate the difference?
Sure. Just give me a minute. I'll just and just give. I'm just searching for the slide.
It's okay.
Yeah.
Take
So,
your
okay. Anyways, how do Sophia solutions compare in noise reduction efficiency? Okay. So, we do have done some studies like, from different competitors, and we do have information for this noise reductions. At present, I have not included those things on the slides, but we do have certain information about that. So it depends upon solution to solution. We can give you we can share you the information separately. What types of studies or sample types, example FFPE, CTT, and low input have seen in most benefit from sulfate in non-specific handling? Uh, what was studies? Okay, so what type of studies or sample types? So we have taken for each, like depending upon the solution, let's just say for the liquid biopsy, we have taken close to 100 or 150 samples to study the noise levels. And like, we do have a comparison studies for each of these solutions. So, and we have observed a lot of noise from the samples, and we tailored our pipelines in such a way so that we can remove the noise and call the variance accurately. So is that answer your question?
So me, are you happy with the answer for the second one? Okay, otherwise just move on to the third one. So
Okay.
is there a quantifiable metric, uh, for example, noise to signal ratio, form
Ah,
positive rate, that Sophia provides user to evaluate the effectiveness of the noise reduction?
okay, so this signal to noise ratio is for some of the solutions we do give the numbers, the signal to noise ratios, but not for every solutions. So most of the times the pipeline detects the noise and like reduces the noise to give you the actual variance. So if you ask me, do we give any specific number for the noise to ratio? No, we do not. We do not give it for all the solutions.
Okay. How about the third one, me? Are you happy with the answer?
Perfect.
Okay.
Thank you.
Thanks a lot. So then, waiting for the other question, I will asking some. I just want to make clear. Um, so what is the difference between the genetic, Sophia genetic and Sophia DDM?
Okay. Sorry, I missed to explain that part. Sophia Genetics is the company name. Sophia DDM is the software that we use for the analysis. We call it as DDM. DDM is nothing but data-driven medicine.
Mm-hmm, mm-hmm. So
Yeah.
and then the Sophia genetic is combine the web lab thing like prompt design as
Mm-hmm.
well as the the patent algorithm for the
Yes.
bioinformatics analyst. And then the DDM also have a lot of bioinformatics software as well, isn't it?
Yes, yes. So in Sophia, we give it as a bundle solution. So where you get reagents to prepare the libraries as well as the software to analyze the samples or the data that you get from this library prep.
Mm-hmm, mm-hmm, mm-hmm. Thanks. And then I saw some slide about the realignment, and I feel very impressive about the result. So,
Mm-hmm.
but we also know that in the get case, the GATK, they also have the realignment. So, what is
Yep.
different about the realignment of pepper compared to the realignment of the GATK?
Uh, can you just repeat the question? Sorry, I missed it somewhere.
Okay. So, can you share your slide again about the slide number around 38 about realignment?
This one?
You did not share your slide.
Sorry, sorry.
Mm.
Just a moment.
Ở đây trong lúc chờ đợi thì mình cũng muốn giống như cái bài nói của bạn này hơi hơi nhiều về cái phần thiên về phần ban phân tích, với lại là không có đi cụ thể vô từng cái chi tiết đó đó. Bởi vậy là nó cũng hơi khó hiểu đối với những người học nhưng mới như tụi mình. À, I just want to asking the realignment here from a
Mm-hmm.
paper technology. So what is different from realignment from pepper compared to the realignment of the GATK?
I'm sorry, I'm not sure about this GATK. I haven't worked on that before. So
Okay.
I am afraid if I'm not able to answer that.
Okay, it's okay. Because
Yeah.
like we also do the realignment step in the best practice of the
Hmm.
GATK Broad Institute software for open source one. Yeah.
Okay.
Okay. Mm.
Hi,
Okay.
hi, Dr. Lloyd. This is Steven.
Hmm.
Mhm.
Yeah, I'm also from Software Genetics. I'm the regional manager for Asia Pacific. So I could answer some of the questions for you. So GATK is a free software that most of researchers use to do the alignments. The
Yes.
difference that Sophia that what our Pepper or our Mocha does is that it's a special algorithm we design and then we run in the background. So, and then we do the alignment using our own technologies, a special algorithm. It's not based on GAT or any of those open source softwares. So essentially what it means that we can provide a better alignment compared to GATK. GATK is an open source, so most of the time you can align with GATK, but for easy ones. But if they're complicated ones, sometimes GATK will miss. And that will essentially lead to the VCF file where you're missing some variant that because they didn't align properly. And what we do with the algorithm that we will be able to have a broader alignments on the segments. So at the end, you will actually get more variants from Sophia alignments. It doesn't mean the more variant you get is more accurate, but essentially, if you misalign it, then you will not find the right variant. And this is, of course, this is proprietary technology developed by Sophia Genetics. It's easier to say than done. So the easiest way to see is that when we share our algorithm with some of our customer, they use some other third-party vendor analysis software, maybe some piece of GATK or their homebrew. They usually see more variant detected by Sophia Genetics, and oftentimes when they confirm by Sanger, that is actually the right alignment. So that is the difference when we do the alignment, do the secondary analysis, where you get actually get more detected variants compared to other software, and this is based due to our Pepper, our Mocha, and also how we reduce the background noises. This answer back to the question from me. How is the noise reduction efficiency? So that's also how we differentiate from the other software company, where we take the background noises on each technology. For example, Illumina sequencing technology, Element sequencing technology, MGI, Thermo. They're all different sequencing technology. They have different background noises And what we do is we understand those sequencing technology, you understand the noises from each sequencers, and we filter them out. And that's how you get cleaner data. And at the end, you get a more higher grade clinical data that you can see used for alignment and for VCF as well.
Yeah, thanks a lot. And then the realignment step, it means that you have your own software to do the alignment, like BWI or Hotei or whatever Dragon using. And I just want to understand the magic step of the realignment, uh, unable detection of the duplication, like the slide of 41. So just want to compare between, do you do any benchmarking paper to compare the realignment step of Sophia compared to the paper here with the other.
Uh, We 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 might have some paper, some of this technology might have some publication, but we will need to look into those ones. But
Mhm, mhm.
some of the core part, usually the proprietary technology is confidential. Even we don't know how our team does it. But
Okay,
what
okay.
we do know is how are the results that when we run these are generally more efficient and have a higher data quality compared to others.
Mhm. Okay, thanks a lot, Stephen.
Problem. Yep.
And then because like in Sudh's slide, he have a lot of new term like about the community panel or community solution. What
Mhm.
does it mean?
Okay. Community solutions are like the custom solutions. So these are designed specifically for for clients who ask like for extra genes that has to be included. So we call them as a community solutions. Let's just say, so for solid tumors, let's just say solid tumors, which we have like the basic panel, which has 42 genes, but some of our clients asked to include the BRCA genes for the breast cancers. So we have included it and we call them as a community solution.
So the community solution or community panel
Hmm.
is for the um, germline detection or somatic detection.
So we do have community panels for both germline and somatic solutions.
Mm-hmm. And then what is the purpose for the panel? For screening of the um, cancer of the uh, Mendelian cancer or the familial cancer, or they use for what kind of purpose, like treatment or monitoring? Um, just want to understand for screening or whatever. What is the purpose for the community chief panel?
You mean what is the purpose of having a community solution, right?
Yes, yes, yes.
It's a panel or a solution with these genes that are required as per your requirement. So that's what we call it as a, I mean, you can call it as a custom solution as well.
So, Satish, I can take this one. So,
Please.
essentially, we call it community panel, but basically, it's a custom panel designed
Okay.
by other users. And
Okay. For any purpose that the user want to do, isn't it?
so, most of these community panel or custom panel is because for different country, different um, territory, different diseases, some institution they have specific genes, a specific target they want to pinpoint, they want to include in the panel. Uh,
Mhm.
some are for reimbursement purposes, some are for drug biomarker purposes, and some are just fitting the regulations that they need to have these in the specific countries. So it's different for every countries. So we have a standard panel that's called a baseline panel, but some other depends on what the lab or the country regulation is. They might need to remove or they want to add a couple more genes in there just so they can get reimbursement from the insurance or just they can fit the reimbursement from the healthcare system or just because these genes are specific to that populations. So that's why we have all these different custom panel. They're based on the customer needs. And what we do is we design these custom panel with the users and validate it with their clinical samples. So once
Mhm.
data is validated, that means this solution it's been proven to work with a clinical sample, and we have the right to sell it to other people other than these users. So that's why we're saying that we have a couple of allow for the community panel where these are designed by other people, but essentially these might be used for some other people than these users, and we are able to introduce these to you, so you don't have to go through the process of customizing and design something entirely new, where this is as a off-the-shelf solution that you can just use it right away and it has been validated.
Yeah, yeah. And then if we have our custom panel gene, and then we lost our CE-IVD, is it? The CE-IVD.
So
Because like,
yes, so CE-IVD and RU are completely different stories. So IVD will require a lot more clinical validations. We submit
mm-hmm.
to the accreditation body to be proven the accreditabilities, the efficiency, robustness, and the results of these specific solutions, along with the sequencer, the flow cell that's being used in the entire workflow. So in a way, it's fixed, meaning it's validated by special accreditation bodies, but it's unchangeable, meaning that you cannot do anything change about that solution. If you do, it's no longer IVD, including the
Yeah,
sequence.
it means that we have the custom
Yeah.
panel, and then
Yeah.
we no longer have the IVD anymore, is it?
Exactly, exactly.
Okay.
And you have to use exactly the same flow cell that's been submitted and approved for the IVD. So if you want to upgrade to the bigger sequencer because of cost, you cannot. The IVD will, if you do that, the solution is not IVD anymore. And in this state, with the advanced technology and all the data coming out for NGS, it's REO is probably the best way to do it because there are new biomarker discovered every year. And IVD, you'll usually take at least 1 to 2 years to get approved. Once you have all the data, remember, these will require clinical data to do some studies and submit it, approve, and then you'll get an IVD. And that's just for the IVD in that specific country. And let's say if we were to enter Vietnam, we have to register in here. If they see there's a need for another study, then we have to do the study before we can introduce to the market. And that's it. By the time maybe the 3 years has been passed by, and there are new biomarker coming in, and so it will be very challenging to keep up, always having the IVD. So in
Yeah, yeah,
NGS, EREO are usually the people most used because you can get the latest panel design and you can use the latest technology. And usually these are usually behind by the drug company, pharmaceutical company. They have new drug coming out. They need a biomarker and they will need some diagnosis tests to run this so the patient can be benefit, meaning they detected having these biomarker variants and they can use the drugs.
yeah, yeah, I understand.
Yep.
Okay, so we have one more question for me.
Sure.
How does Sophia genetic handle genomic noise from the region like homopolymers or highly GC-rich sequence? Are there specialized algorithm for this?
Yes, so I will answer this question. So the Sophia DDM pipelines or the algorithms that we have are like this can detect the homopolymers as well as the high GC rich sequences. It will differentiate between the noise that we get from these regions, and it will filter down all those things, and it will show you the true positive variance. So if you ask me how we do it, we do not have the information as this is a proprietary thing. Yes, we do have specialized algorithms to look at this homopolymer and high GC rich sequences to filter it out.
Okay, so the algorithm we just have the name, is it? The pepper.
Pepper, muscat, all these are the algorithm names which
Yeah,
are
yeah.
presented.
Thanks a lot, Sudesh. Can you
Yeah,
come back to slide 35?
35.
Yes.
Okay, just give me a minute. Yes,
Okay. So if I by myself I look into the 2 nanogram, and it
hmm,
look like over here have a lot of duplication
yes,
because the read is exactly starting and ending at the same position.
yes,
And So for the 20 nanogram, for me it look better.
Yes.
So and then what is the I'm missing the slide take home message here.
Sorry?
I don't understand. I get lost here. I don't know what is the purpose of this slide.
Okay, so I'll tell you when you have when you use a 2 nanogram input, what you
Mhm.
do is, like, to amplify the product, you need to increase your PCR cycles, which will
Exactly.
increase the duplication rate. But when you
Mhm.
have a high nanograms of input, like in a higher input, you don't need to go with the more number of PCR cycles so that your input, the sequenced molecules decrease the chance of missing variants. And it is the molecules are like highly diversified across the regions.
Yes, and then you come into the 36 and what does it mean?
Okay, so when you have lower inputs, you can see
Mhm.
there are like more false positive. This is because of the noise levels that produced because of the lower input materials. But when you go to the higher input materials, you can see the variance can be called with confidently, as you can see the noise levels are less. So this is because of the input material like that you have taken. Lower the input, there are more chances of getting a false positives here because of the noise that we get from this lower inputs. But when it comes to the higher input, the noise levels will be reduced and you can call the variance confidently.
Yes. So and then I just want to understand that the Sophia genetic,
Mhm.
what it can have here in this the situation. Like we have low input, but we want to have the low form positive or form negative.
Can you repeat the question? Sorry.
So Dr. Loy, so the message we have for these two slides is essentially, Sudhir, can you go back to the other slide, 35? So this slide, the message is that for some people, they might think that if you have less DNA, you just double or triple the amount of the PCR cycle, then you'll get the same amount if you have 20 nanogram. But the fact is, even if you double, triple, you get a lot of the same DNA content, but just in lot more volume. You don't have the diversity because your starting material is too low. So the message we want to deliver here is that even though yes, you can get the same amount of DNA, if your starting material is small, but you won't have this right DNA diversity, meaning you won't have all these different colors you see on the right side. So the best way to have a good quality of data is you need to start material as much as you can. That is the first metric from the 2 nanogram and 20 nanogram. That is a difference from this side. You can see that if you can start more DNA, please start more. Don't start with little DNA. That is only if you don't have it. And that will be the challenge you see on the next slide. If you start small amount of DNA, then basically your diversity is very small, meaning you have a lot of duplication on the same fragment. And that will introduce a lot of noises, and you can see that the true positive will be generally very small. And compared to if you have a higher amount of starting material, let's say from 20, 30, or 50, you will see generally you have less background noises, but more true variant because the signal level is much less. And that's the message we are trying to share with people that we've seen a lot of data. People say we don't have enough, we don't have a lot of DNA, so we just duplicate the amount of the PCR cycle. That would not help. The data would not help you because you start small, then that's as much as you can get. So the best way is to start a right amount of DNA, then you have a very good data because if you don't have good material in the beginning, it's very challenging. It doesn't matter who does analysis, they still have difficulty finding the variant with a confidence level to give you that this is the true variant or this is actually background noise or this is true, this might could be the false positive.
Yes, understand it. I just don't see that what is the Sophia genetic over here rescue the situation like we only have 5 nanogram, but we want to detect the good rate of the variant detection.
Yeah, so in the FFP sample, so one of our um technology that is that we call it group readings. So what we do is that we tag the same fragment in couple points in each of the fragment within our a library prep and also algorithm. So if the same fragment should have three taggings, but maybe the other four or five strand only has two. That means those are not the real ones. So we have our technology. So even though if it's starting low, we can still help you to get good quality of data. But the best way is always having more starting material than lower DNAs.
Mm-hmm.
Yeah. So that's something that we do can help. But of course, if it gets very, very low, it's still very challenging on analysis part. That's we're trying to
Yeah.
deliver the message to the general people when people starting doing the NGS field.
Yeah, thanks a lot, Stephen and Sudesh. And
Yeah.
then I know that this is the know-how, the secret of the Sophia, and then I will check one curiously. Want to understand the uniform correct by block design. What is the magic here to make it very uniform coverage of the sequencing?
Yes, the uniformity.
Yes, yes. Here, no, yeah, yeah.
This
Not
one, right?
exactly this one. I think that this one is,
This one. Yeah.
yeah. Where the magic come from?
The Sudesh can elaborate more in detail, but from in general layman term, basically this is due to the probe design. When the vendor designed the panel, designed the probe to target that specific genes. If
Mm-hmm.
he didn't have a good design, you will see a fluctuation on some of the part on the exome. So if they didn't have good enough coverage in that specific area, then if you want to maintain the same coverage, meaning the same coverage, same reads in that specific area, you have to increase your sequencing depths just to cover that enough. But if you see from this slide, in our design, it's very uniform, meaning there's no up and down, right? So they're all very uniform, meaning that you will be able to, it's basically due to the probe design we put in there to make sure we have enough coverage for these three genes, and these are very challenging genes. If you look at other vendor, either Amplicon or L Capture, you can see they fluctuate a lot, meaning that
Exactly.
some place they need more they need more read to cover. And that means you need the same flow cell, you need to take more space just for that specific genes. And at the end, that means you can only sequencing less samples. Whether you have a more coverage, a more uniform coverage, then you're actually sequencing more samples. And that at the end, it's actually helping the whoever's running the sequencer to save the cost because if you can sequencing 10 sample, but because don't have a good uniformity, you can only sequencing 8 samples. Your costs go up by 25%, right?
Yeah. So the point is over here. Just want to asking that we in order to do the to have the uniform coverage, then mean we have to design more prop to cover more region or
Yes, that's the general message, but usually if
Mhm.
it's by the third party vendor, you have no control on how they design the probe, right? So you won't know until you run it and see, okay, these are the coverage. And but if you were to design a panel yourself on the coverage, you have to take into consideration how to design to make sure these are coverage is enough. And this could take experience because we have a very experienced bioinformatics team in the company in Switzerland. So they know how to design, they put this and they test it out. So that's how we can achieve this with a very good uniformity. And then if you look at some of the company that you see, they can give you a good result, but then you can see their sequencing cost is much higher because some area they don't have good coverage. And that means that if you detect something a specific variant mutation in that region, you will have very low risk. That means you can be confident it's actually a true variant or it's a background noises, and that's where you don't want to happen. And then if you were to design it, you have to really think about how to design this because we know in some cases by adding or removing some gene together, the same probe might not work because they interfere with each other. So these will take experience to actually know how to design this.
Mhm. Mhm. And sorry that I asking too much. It look into it, and then we can see that there is the forward and the reverse trend is imbalancing. Like for example, in the gene FLT3, on the left hand side, we saw that the red color is much more on the left hand side compared to the right hand side. So I don't know that this one is the full coverage of the whole thing or not. So for me, if we look into it, it looks very biased into the forward trend on the left hand side.
On the yeah.
It's okay. It's very detailed. Sorry. Okay. Thanks a lot, Stephen and
No
Suresh.
problem.
Yeah, thank you.
Có ai hỏi gì nữa không, mọi người? Có ai có thắc mắc gì đó không mọi người? Mình giúp tóm lại tiếng Việt để cho mọi người nắm một tí xíu thì công ty Sophia thì cũng cung cấp những cái giải pháp về bioinformatics, phân tích bioinformatics cho ung thư. Mọi người cũng thấy đó, thì ở đây cũng phân tích về liquid biopsy, về solid tumor đó, cho solid tumor với lại là cho cái ờ, liquid tumor giống như những cái ung thư máu đó. Và có những cái giải pháp từ đầu cho tới ra cái report, cái report mà đưa cho bệnh nhân luôn mọi người đó, từ đầu đến cuối. Và có những cái giải pháp là có IVD đó. Dĩ nhiên là không phải giải pháp nào cũng có IVD hết, đặc biệt là hồi nãy tôi cũng có hỏi là có những cái mà custom panel á, thì mình đưa vào thì nó sẽ mất đi cái IVD của một số cái giải pháp mà có sẵn đó. Tại vì người ta làm nó đúng theo chuẩn, bây giờ mình muốn thêm hai ba gen vào hoặc là bớt đi hai ba gen gì đó thì nó sẽ mất cái IVD đó. Thì ở đây có những cái giải pháp của Sophia, thì trong đây là Sophia Genetics và cái Sophia DDM đó. Mọi người cũng thấy là có nhiều thuật toán, có nhiều cải tiến để giúp cho việc phân tích nó tốt hơn, cho kết quả nó chuẩn chỉnh hơn đó. Nhưng mà ở đây thì tôi cũng hỏi, và có bạn B cũng hỏi nè, đó có nghĩa là công ty của người ta thì người ta chỉ nói người ta làm được như vậy đó, còn làm được như vậy có đăng công bố hay không thì cái này thì mọi người cũng thấy là cái này bí mật đó của công ty của người ta đó, người ta cũng không có trình bày cho mình cụ thể đó. Có những cái ví dụ như giờ ai người ta thiết kế cái prompt thì người ta cũng biết được là thiết kế cái prompt cái tiêu chuẩn nó như thế nào, đúng không đó. Nhưng mà riêng ở đây mọi người thấy là người ta cũng thiết kế cái prompt mà cái độ phủ của người ta rất là đồng đều đó. Thì mình hỏi thì người ta cũng trả lời nhưng mà nó không có cụ thể, đúng không, tại bí mật mà, đúng không đó. Kinh doanh mà người ta đâu có nói cho mình nghe cụ thể. Mọi người cũng thấy đó, ai thiết kế chứ còn người ta thiết kế thì sẽ ra đều, còn những người khác thiết kế thì không đều đó, không đều bằng đó. Mọi người tự nhìn thấy ha, cái slide 30 này đó. Còn bí mật như thế nào thì cái này kinh doanh của người ta mà, đúng không, ta đâu có nói cho mình nghe hết đó. Rồi, không biết là các bạn có hỏi thêm gì không hả? Rồi không hỏi tôi hỏi tiếp nha, cho quyền hỏi không chịu hỏi đó thì tôi lại hỏi tiếp, để nói tôi giành chiếm sống. um, can you come to the slide 45? I don't understand the A B C D classification.
Slide number 45, right?
Yes, number 45.
This one?
Yes, this one. So for my curiosity, I just want to understand what is the A B C D classification.
So these are the classifications that we give on the platform for the tertiary analysis on the Alamut Visual Plus. So this is the basically the classification that show on the software. internal classification, I would say.
So it will classify the variant, isn't it?
Yes.
It classify like the benign, slightly
Yeah,
benign.
okay. So I'll tell you in detail way. So this is how
Mm-hmm.
we classify it. When we have a variant which is pathogenic, so we call it as A, which is pathogenic, and
Mm-hmm.
B is likely pathogenic, C is VUS, and D is benign. So this is how we classify
Okay.
with the Sophia filters.
Okay. 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 Thanks a lot. And uh, what algorithm the A, B, C, D are using? Because like we have the AlphaFold, we have so many algorithm available. And
Mhm.
this one is from your company, Sophia, developed.
Okay. Okay, I understand this. Like, how we give this classification for benign. So we do have data sets from different clients or like, across the globe. So based on their inputs as well as the AI that we use to get this classification with the Sophia filters on the DDM. So algorithm, I'm not really sure what algorithm they use, but we use an AI model and the inputs that we get from the global database across the client.
Mm-hmm, 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 mm-hmm. Thanks.
Yeah.
And the ACMG score. So
Mhm.
you already classify them, and then you also calculate the ACMG score as well.
So, yeah, on the tabular, like when on the DDM, we'll give you the classification from Sophia as well as the ACMG score in the tabular form so that you can compare those. So the variants which might be pathogenic in one of the regions across the globe will not be pathogenic in a specific cohort. So we'll give you both the options so that you can check according to it.
Mm-hmm, mm-hmm. And then this ACMG score, do you compare with the VASM database just to make sure that it's the similar scale or
I didn't get the question.
so just want to understand that the ACMG score over here is
Hmm?
equivalent to the classification of the VASM ACMG.
Varsum ACMG.
Yes.
I'm not sure. I haven't used the Warsome, so I
Okay.
can't comment on that.
Okay. Okay. It's okay. It's okay.
Yeah.
Thank you.
Yeah, so they it's different. So it's they have their own technology to the classification. We have ours, so they
Mm-hmm.
are different. So yeah,
Thanks,
Okay.
Steven.
yeah.
Okay. Thanks. Thanks for this.
Yeah.
Um, any more question? Okay. If no question more, thanks a lot, Sudet and Stephen, that help us to understand about the Sophia product and service. Thanks so much. Uh, it's very informative and have a lot of new things that I learned today. Thank you so much, Sudet.
Thank you. Thank you so much. And I'm glad to hear that. And thank you so much for giving me this opportunity to present about the Sophia Genetics on today's call. I really appreciate it. Thank you.
Yeah, thank you.
Thank
So
you,
and
everyone.
then we will post in our YouTube, and then
Mhm.
a student can review the lesson, the your presentation when they have time. Thank you so much.
Yeah. Thank you. Thank you so much.
Okay, thank you, Thomas. Thank you, everyone.
Thank you, everyone, for joining. Bye bye.
Bye bye.
Bye
À,
bye.
cảm ơn mọi người rất là nhiều ha đã tham gia buổi hôm nay. Buổi nó cũng ngắn nhưng mà có nhiều cái học hỏi rất là tốt ha mọi người ha. Đó, mỗi công ty sẽ có những cái riêng đó vậy đó, và mình học được cũng cái này cái kia để mình có thể tự so sánh được ha. Cảm ơn mọi người rất nhiều, chúc mọi người buổi tối ngủ ngon chào mọi người, bye bye and good night, see you.
Yeah. Bye bye.
Thank you.
Thank you.